All right, we've hit the halfway point and we are ready for our third presenter. I'm going to introduce Mr. Li Zhou from the Department of Agriculture and Bioresources in the Department of Plant Sciences at the University of Saskatchewan. Those of us who have flown into Saskatoon knows it can be pretty frigid during the winter. So kudos to you for not only doing research, but tolerating the chill up there. And here we go.
Okay, yeah, so good afternoon, everyone. My name is Zhou. So today I'm going to show part of my research, electrostatic treatment as a novel and efficient method in vegetable oil refining. So as we all know that the canola vegetable oils, when they are extracted from the plant seeds, they're dirty. They have various types of contaminants inside. For example, the free fatty acid, the phospholipid, the pigments, peroxide, those contaminants negatively affect the quantity, the appearance of the edible oils. So those contaminants are usually should be removed from the edible oils. The conventional vegetable oil refining method, they have some drawbacks. For example, they're energy intensive and they cause the substantial a huge amount of nutrient oil loss. So the new vegetable oil refining method are all they are required to be developed. Then there are a lot of reports regarding the cleaning of industrial oil. They use the electrostatic field, and it has been successfully applied in the industry. They are very good for the industrial oil, for example, the lubricating oil and or some other industrial oil cleaning. So we have an idea that we can use the electrostatic treatment to refine, to clean the vegetable oil. So that's the background. Then we did some research through a commercial electrostatic device to clean the canola oil. So the objectives including to evaluate the effect of the electrostatic field to remove few types of contaminants, free fatty acid, phospholipids, pigments, and peroxide. Also, we want to make sure that if this new method will cause the damage to the triglyceride structure, to the oil structure. Also, we have tested the nutrient oil loss during this process. So they are the objectives. Now the materials method. As I mentioned, the research was conducted through a commercial electrostatic device, and the raw materials is crude canola oils. So the crude canola oil in the container pass through the electrostatic field repeatedly, and the samples are connected at a different time. And then the samples are analyzed. And two different types of experiment were conducted. A bench sky is only 1 liter, and the pilot sky is 15 liters. Then the main test, or the phospholipid content, which was conducted by NMR. Then the pigments and the free fatty acid peroxide are tested for the related AOCS method. Now let's move to the results. Firstly, as we can see from the figure 2, in both bench scale test and the pilot scale test, there was a significant reduction in the phosphorus content in the canola oil after the electrostatic treatment, which means that this method works well on the removal of phospholipid. Then the acid value is presents the content of the free fatty acid. The results also show that in both bench scale and the pilot scale tests, the method works well on the removal of free fatty acid. Then the next one is a peroxide value. Peroxide value means the content of the peroxides. it looks like the content of peroxides reduce gradually, but still significant reduction can be observed. Then the last one is beta-carotene. It looks like the effect on the removal of the beta-carotene is limited compared to the removal of other contaminants. Now the next part is a study of the free fatty acid. The results show that where I actually just show the main fatty acids, the palmitic, oleic acid, neonic acid, and neonic acid. The results show that there was no significant change in the fatty acid composition.
We're coming up on the 5-minute limit, so I'm going to ask you to wrap it up.
Okay. So the result shows that there was no significant change in the fatty acid composition. Then the last one is a neutral weight loss. The results indicated that the neutral weight loss is only 0.37%, which is very negligible compared to the conventional weight refining method. So the conclusion is that this method works well on the removal of phospholipid, free fatty acid, peroxide, and it works on both different scales, and it didn't cause the damage on the fatty acid profile. Also, it caused a very negligible neutral weight loss. And finally, I would like to express the appreciation to the Saskatchewan Ministry of Agriculture for their financial support. Okay, thanks.
Thank you. Okay, well, I'm going to check the community for some questions. I'm not seeing any, so I will hand it over to the judges. Have at. We have 5 minutes of questions.
Okay, I'll go first again. This 0.37% of losses. This is we're talking about oil that you wanted to keep, basically. So there were other losses in terms of the fatty acid or the phospholipids, but the 0.37% was a loss of oil, like neutral oil you wanted to keep. Is that correct?
Yes. Because okay. Because in the conventional vegetable oil refining, they typically is from the literature reviews, typically costs 2 to 5% of neutral weight loss. So one objective of this program is we believe that this method will cause much less neutral weight loss compared to the conventional method.
Okay, but I'm not sure, we haven't produced an oil that is of a full edible quality, though, have we, just by this method? It's kind of like a pre-treatment method. Wouldn't you say that's what it is? Or have you really produced an oil of a fully deodorized, etc., RBD quality?
Oh yeah, I agree with your opinion. We can treat it as a pre-treatment because it can okay. From the phospholipid free fatty acid peroxide removal rate is only like 50%, not like 90%, 95% compared to the conventional method. So yep. Yeah, so it only can be acted as a pre-treatment method.
And do you think it's scalable up to a large, if you had hundreds of tons a day? Is this a feasible in terms of power consumption and size of unit? Is it is there a way to sort of scale that up to an industrial level?
Okay, there are some limits. That I didn't mention in the poster. The first is actually there's a limitation in the moisture content of the oil. The moisture content is actually 500 ppm. So to make it for the industrial production, we need to make sure that the moisture content of the canola oil is low. Otherwise, we need to put some pretreatment, for example, the dehydration process on oil. And besides, the voltage is actually very high. It's as high as 10,000 watts. So it's also, dangerous. So there are some other work need to be done to solve these problems.
Yeah, okay. I mean, 3,500 parts per million is quite dry. It's not it's not unreasonable, but yeah, you could easily have oil with more water than that if you just took it as crude.
Yeah.
I want to follow up on Alan's question, or I would say more of your point that you said, moisture content. Wouldn't the electrostatic forces remove the water content in your oil at the same time?
Pardon me, can you say again?
Your electrostatic forces, your electric field, would it not remove the moisture or any water in the oil?
Oh, I did not test that. Yeah, but some from the literature, some people use that to remove the oil. To remove the moisture from the industrial oil.
Okay.
Yeah, so it
Thank
should
you.
be work. Yeah, but
Okay.
that's
And
not my point for the edible oil. So.
Okay. Now you're saying, you're talking about scaling up things. You said moisture content would be a problem, right?
Yeah.
That's what you were saying earlier. So
Yeah.
if the electric field can remove the water or moisture, that can solve the problem, right?
Yeah. But how do you say? Okay, in the industrial oil, some people add some additives. They put additives. The additive will mix with moisture together first, and then removed by the electrostatic field. Then, in my research, if moisture content is very high, it will cause the short circuit or something. So that's why I did the dehydration process to reduce the moisture content to very low.
Okay.
Yeah.
And I have one more question regarding your phosphorus data. You said you've used NMR to determine the phosphorus content, right?
Yeah, phosphorus content. Yeah.
So what is the internal standard that you use for your NMR analysis?
Pardon me? What's the what?
What is the internal standard that you use for your NMR data analysis?
Oh, okay. Just give me 1 second. I cannot remember the name of the chemical. It's called a triphenylphosphine.
Triphenylphosphine.
Yeah.
Okay, thank you. So what's going on in the graph? If you have your data with you, just look at it and explain. You have differences between the pilot scale and the bench scale data.
Yeah.
It's kind of weird trend that the phosphorus content goes down, where you're showing the rate of removing phosphorus, shoots up and then it falls back down, and you can see the phosphorus content go back up. Have you looked into what's going on there? Like over the time, you saw the phosphorus content come down and then it goes back up. Could you explain on that?
Okay. Actually, I feel that the most reduction in the contaminants happened at the first, first time when the oil passes through the electrostatic field. Even in some samples, the content increase a little bit, but yeah, I still feel that the most reduction was happened at the first time. First time it pass through.
Okay.
Yeah, looking at figure 2 on the poster, there is quite a sort of what I see initially the big fall in phosphorus content, and it kind of creeps up to 60 minutes, and it goes down again at 390 and 450.
Oh, that's two tests. It's the 10, 20, 30 minutes. They are the 1 liter scale, the bench scale, and the 60. 740, 450, they are another types of.
I see.
Yeah, so they're two different tests.
Oh, but you still got, it seems to be going up at the bench scale test. It's kind of rising 10, 20, 30. Or is that just error?
Yes. I just guess there may be some sample error or testing error or something. I still believe they should be reduced. The most the phospholipid are removed at the first sampling.
So
Yeah,
I think
there's a
we have
big
time for
jump.
one more question. I don't want to let a process engineer from DeSmet miss a opportunity to ask a question when neutral
couldn't
oil
have
loss has been mentioned. So
Very much
Rain, do you have a question?
Very much appreciated, Michael. Did you look at the difference between if they were hydratable and non-hydratable phospholipids that the process was removing?
Oh, sorry. Yeah, can you please repeat?
Yes, sir. Did you note when you were seeing the removal of the phospholipids, if it was removing some of the hydratable phospholipids versus the non-hydratable phospholipids?
Oh, yeah, that's a good, very nice question. That's actually the problem of the test method. Is the test method is I use is NMR. Actually, the NMR can show, the different type of phospholipid, like I don't know name, but PA, PI, PE, right? Because there are different types of phospholipid. So currently, I just test the total phosphorus content. I have a book the standards of the different types of phospholipid, like a PE, PI, but I have not tested it.
Okay, I really enjoyed how it was laid out in one place, and I could see all the information there. I would really, very interesting stuff. I'd love to see more information on that as well, and as well as ratios between hydratable and non-hydratables and how this affects that.
Yeah,
so thank you very much.
Yeah, the NMR needs the standards to test those.
Yeah.
So
yeah. Very interesting presentation. I think that's the first time I've seen phosphorus analyzed by NMR. I'm used to kind of seeing ICP results in the mornings from the plant. So thank you, Lee.